now. So in other words, on Friday, be able to answer questions on how prokaryotes do transcription in terms of what their promoters and their terminator sequences, how eukaryotes do transcription with transcription factors and RNA polymerase II and polyadenylation signals, and know all about how the pre-mRNAs in eukaryotes are processed, like the 5' cap adding and the poly-A tail, and all about the introns, which is this topic that I think we finished off on. Yes?
You said that the viruses and bacteria and going to be for last, is that
wait, no, so I'm saying that the next topic I'm going to cover in here as soon as we finish genes are viruses and prokaryotes. But when you come to the laboratory, well, actually, just bring with you at all times, both in the lecture here and in the laboratory, the handout on DNA technology, because I'm going to start covering bits of that both here in lecture and in the laboratory setting. Okay, well, yeah, so let's do a quick review of introns. Here's this diagram. I guess I showed you this last time. If you, this is supposed to be a messenger RNA, a pre-mRNA, as it's just been rolled off, as it's just been transcribed from the gene, and the protein up above is the protein that's supposed to be made from this particular RNA. And if you look for the codons that match up with these amino acids, like here's methionine, its codon is AUG, and here's arginine, its codon is CGU, and lysines is AAG. If you look for those codons, you can find them in the RNA, of course, but they're not continuous. The codons for the first 3 amino acids are there, then there's a bunch of G's, A's, U's, and C's in the RNA that don't encode any amino acids. Then you find a couple more codons for the next 2 amino acids, and then there's a bunch of G's, A's, U's, and C's that don't encode any amino acids, and then there are the last 2 codons right there. These parts of the original RNA transcript that do have codons, that do get translated into amino acids by the ribosome, are called exons, and the parts that do not encode any amino acids are called introns. And the introns have to be spliced out before this RNA can leave the nucleus, because if they weren't, the ribosome would think that these are just more exons and start making, or just more codons, I should say, and start making amino acids based on these sequences, and those amino acids are not part of the protein that this messenger RNA is supposed to represent, is supposed to make. And just to sort of underscore that fact, I printed out a little pretend messenger RNA here, or I should say a pretend pre-mRNA. And so it's all just G's and A's and U's and C's. And so there's nothing in and of itself that makes the introns different than the exons. They're all just G's, A's, and U's, and C's on the pre-mRNA. To splice out the introns, there are proteins inside the nucleus that look for specific sequences. There are certain sequences along the pre-mRNA that says, I'm the beginning of an intron, and other sequences that say, I'm the ending of an intron. And by keying in on those particular sequences, there are enzymes, there are proteins inside the nucleus that cut out the introns. We've seen this before. The introns get looped downward like that. The introns are the pink things, and the exons are the gold things. And eventually these enzymes just physically cut out the introns and splice, that is to say, ligate the exons together. And here's a cartoon. The proteins that glom on to the beginning and the end of the intron are called small nucleoribosome term. I have to cheat here. Small nuclear ribonucleoproteins, or SNRPs for short. That's how I remember them. And as they start to come together, they loop out the intron. And when all the SNRPs from the beginning of the intron and the end of the intron come together, like you see there, that whole cluster of enzymes is called a spliceosome. And the spliceosome physically snips off the intron bit and ligates the exons together. All right, sound good? Sound familiar? Okay, now these, the introns that you find in the pre-mRNA, which are the red parts of this diagram right here, like there and there and there and there and there, the introns are found in the pre-mRNA because those intron sequences are also found in the gene that makes the pre-mRNA. So if we're trying to understand, well, why do we have introns in the pre-mRNA? Well, it's because you find those same intron sequences in the gene itself. And so the question really is, why do eukaryotic genes have intron sequences in them? They're not necessary because prokaryotic genes do not have any intron sequences in them. So obviously you can have a gene that doesn't have introns. And people have puzzled over why eukaryotic genes have these things. And there's a couple of reasons. I think nobody knows really for sure, but there are a couple of reasons, and they both relate to this fact right here, that exons usually correspond to functional domains of the protein. And I gave this as an example. If you think of a protein like the tyrosine kinase signal molecule receptor, different parts of the protein have different functions. Gesundheit. The part towards the outside binds the signal molecule. Oh, okay, yeah, they have to dimer. Just see that. There we go. The part towards the outside, the extracellular portion, binds the signal molecule. That's the ligand binding domain. And the part that's in the membrane anchors the receptor to the membrane because that's a hydrophobic domain. And the part towards the inside is the part with enzymatic activity. It binds ATP, so it has an ATP binding site, and it also has a site where it takes phosphates from the ATP and attaches them to tyrosine amino acids. And so it's one protein really, but the different zones of it, the different domains of the protein have different functions. And what they found when they analyzed genes is, in general, each domain of the protein is encoded by its own exon, or at least by a small group of exons. So, for instance, the first exon of the gene for this protein might encode the ligand binding domain, and the second exon, the hydrophobic transmembrane domain, the third exon, the intracellular domain, that where you find the ATP binding site. So, I guess I would say that these introns then divide up the coding region for the protein into different zones, and they're modular in the sense that this exon encodes a small segment of protein with a certain function, and this exon encodes a small segment of protein with a certain function, and this exon encodes a small segment of protein with a certain function. And so why is it helpful if our genes are modularized in terms of the protein regions they encode? Well, there's a phenomena called alternative splicing that takes advantage of this modular arrangement of the parts of the gene that encode the proteins. From what I've been telling you so far, from what I've been explaining how this works, I've said that this exon right here ends up spliced to the next exon down there, and the intron between the two gets spliced out by the spliceosome. And that happens, the intron there gets removed. And then likewise, if this is exon number 2, it gets spliced to exon number 3, and that intron right there between the two gets removed. So far as expected, and likewise, the next two exons get spliced together. Fine. But what they find is, in some cases, an exon doesn't always have to be spliced to the next exon downstream. Sometimes the spliceosome could take this exon right here, gesundheit, and splice it to this exon right here. In other words, this whole segment right here, including these two introns and the exon between them, can end up being spliced out like that. And so thanks to this modular arrangement of the exons, you can get more than one protein out of the same gene. Because of course this is a pre-mRNA, but it came from just one gene. But by deciding which exons to keep and which ones to cut out, you can get different proteins out of this thing. And the name for that phenomenon is called alternative splicing, when an exon has the option of being joined to more than one downstream exon. And let me give you a real-life example of this. When I was a graduate student at UC Santa Cruz, part of my thesis research involved investigating this alternative splicing phenomenon. The laboratory I was in was studying actually a receptor protein called the mouse growth hormone receptor protein. And it's actually a protein within the tyrosine kinase family of receptors. This protein sits in the membrane of some mouse cells. It has a binding domain for growth hormone. It has a hydrophobic transmembrane domain and an intracellular domain for cell signaling. But what we've also found in studying the mice, that there was another protein that looked like this. This was called the mouse growth hormone binding protein, where this one over here was the mouse growth hormone receptor. Now, this growth hormone binding protein did not have any hydrophobic transmembrane domain, because if it did, it would be anchored to a membrane somewhere. This actually has a little hydrophilic part we call the hydrophilic tail. But the actual hormone binding domain was exactly the same amino acid for amino acid as you find in the receptor. And so it seemed likely that these two proteins were coming from alternative splicing of the same gene. And my thesis research centered around cloning the gene for the growth hormone receptor to see if the binding protein was being produced from the same gene by alternative splicing. And it turned out that that's exactly what it was. This is the structure of the gene as it emerged. It turned out to be kind of a large gene with lots of exons. But here we go. Exons 3, 4, 4B, I'm not sure why we came up with that name, 5, and 5 together encoded the growth hormone binding domain. I guess 6 and 7 also. It's been a while since I had this thesis, obviously. The exon right after exon 7 encoded the hydrophilic tail of the binding protein, whereas exons 8, 9, and 10 encoded the hydrophobic transmembrane domain and the intracellular domain. And so what we found is, what it turned out to be happening here is that both the receptor and the growth hormone binding protein in mice used these exons right here, 3, 4, 4B, 5, 6, and 7, to make a growth hormone binding domain. And then in some cases, exon 7 was spliced to exon 8A, which would then make the growth hormone binding protein when that RNA was expressed. And in some cases, exon 7 was spliced to the other exon 8, this one down here. And in other words, exon 8A was cut out. And so then, since exon 8 is the hydrophobic transmembrane domain, you got one that was anchored to the membrane and followed by exons 9 and 10, which were for the intracellular domain. This alternative splicing is not a real isolated rare phenomenon. What they found is that in fact, in the majority of genes that they look at, there is alternative splicing going on. And to put some numbers to this, remember that humans have around 25,000 genes. And so at first you might think, well, we must have 25,000 proteins or so inside of us. But the number is a lot larger than that because, as I said, a lot of these genes can undergo alternative splicing. And in some cases, you can get not just 2 different proteins by alternative splicing. There's lots of different possibilities. So you might be able to get 3 or 4 or 5 proteins out of the same gene via alternative splicing. So the number of proteins in human beings, they don't know exactly. I've heard estimates from 50,000 to 100,000 proteins inside us. Nobody knows for sure. Any questions on alternative splicing? Okay, so maybe if I just summarize alternative splicing in a slightly different way, I'd say alternative splicing allows for more than one protein to be made from the same gene